Good morning, everybody. Thank you, Betty, for inviting me. And thank you for the organizing committee. Can you hear me okay? Do I speak loud enough? Okay, so it's a little bit an official announcement today. This is actually, Evergreen is the first place where we are going to talk about that. Indeed, there are some changes taking place in Europe. And recently, we got a grant for the first multi-center clinical trial that is going to take place, and the project has just started. Okay, so about the call, it was a call which was open last July. It targets specifically drugs and vaccines for infections that have developed or are at risk of developing drug resistance, so we're exactly in the subject. There were 78 proposals submitted for a first step of selection, 20 that remain after that, and we were very happy in February this year to get into negotiation with the European community. So this is the ratio of type of projects that have been funded, and we suspect that we have this one here with alternative methods. So overall, the project is a 5 million euro budget, and Europe is funding close to 4 million. Oops. Okay, so as I said, the work started on the 1st of July. It's a two-part project. First of all, for a year, we are going to produce GMP phage cocktails, good manufacturing practices. So in other words, therapeutic products that fit the regulatory issues, the regulatory constraints. And then the second part would be the clinical trial. Recently, since the beginning of August, we just opened a website where all the details are being already explained, and there will be regular information on that website to explain how the things are progressing. So here's the project. This is actually, we are going to target wound infections. We have two products that have been prepared. One is against E. coli infections, and the second one is Pseudomonas aeruginosa infections. So the main participants are all listed here. Let's see if I can use this thing. Here you have the, as coordinator of Pherecydes Pharma, and the Pharma unit. We have another important site, which is here in Lyon, in France, which is another burn unit from Hôpital Saint-Luc Saint-Joseph. In Switzerland, we have the burn unit of the University Hospital from Lausanne, as well as the University of Lausanne, who is involved. We have also another burn unit of a University Hospital in Nantes, in west part of France. And then we have also 3 centers in Belgium, including the Queen Astrid Hospital that you know very well, for some of you that are involved in phages. And then you have 2 other burn units, one which is called L'Overval Hospital, and the last one is the Liège University Hospital. And I should mention also as well that, of course, we have Jérôme and his patient association, and also here CRO, which is helping us to coordinate all the clinical activity of the trial. So here you have the details about all the persons involved that I already gave, with the contact person if you want to get in touch with them. Here I would like to make a little bit of a statement, because I think that's the only way we can go around. Certainly I am representing a private company with, in the future, profit points of view. But I must admit that all this work has been and is only possible with this type of collaboration. And here the collaboration is clearly a mix of military, public hospitals, private structures, research, university, everybody in the same boat. And the way we see things in Pherecydes Pharma is that we are very much open to collaborations and sharing information. I don't think a small company like ours, and I don't think there are enough big companies in the phage business to work on your own and to do the things on your own. So this is a little bit the work package design. So during the first 12 months, we are working into setting up two clinical batches under GMP production. For that, we have a company which is also based in France. Its name is Clean Cells. That has a GMP production suite, biosafety level 2 and biosafety level 3. We have been working for the last year to set up already a pilot production process, and we are in the phase of transferring this pilot production process to their facility to get the product produced. Then, meanwhile, we're also working very actively with the authorities and actually meeting the European deputies, meeting the European Medical Agency, meeting the national agencies, because in Europe you have the Central Regulatory Agency, but each country has its own national agency, and you have to coordinate all this together. Because we want to create a new status to bacteriophages into the European pharmacopoeia, and not stick only with a product that is going to be fixed as with antibiotics. And there is a slot to put the products in that type of category, but it would be really a mistake. We believe that we have to have a new category where you need to add the possibility of the product to evolve and to change, like it is already the case today for the vaccine against flu, for instance. You probably know that every year the valence of the vaccine is changing. So we want to get them to agree that this is going to be a moving product, that you have a fixed cocktail, but that you can switch and change some of the phages in the cocktail, maybe every 2 or 3 years, when resistance takes place or when new strains are coming up. Just remember one thing. About 3 years ago, nobody ever heard about the NDM1 E. coli strains coming from India and Pakistan. Now these strains came from India and Pakistan to UK through the people working in UK and going back to see their families. And when that NDM1 strains come into an hospital, it's panic. But 3 years ago, we would have had no phages against that. Now we have some, but this is exactly the situation which demonstrates that you have to have an evolving product. So then the third package will be the clinical trial itself, with of course analytical biology labs from hospitals involved into analyzing the strains. We are going to analyze the evolution of the phages in the meantime. Then there is all a part with statistical analysis, and we already prepared the protocol trial with clinicians and statisticians to make sure that we have something which has the proper format with good significance. And then there will be all a part about writing up the data and publishing. And I'm almost finished, I hope. So these are the measurables that we have put in the project. Well, I also asked the participating hospitals to check their last 3 years record of multi-drug resistant cases in E. coli and in Pseudomonas aeruginosa. And actually it was a bit of a surprise for us because everybody talks for wound infection about Staphylococcus and Pseudomonas, much less about E. coli, but actually when we look at the statistics from the last 3 years, we end up almost having the same level now of E. coli infection as of Pseudomonas infections, and especially due to the rise of these new ESBL resistant form, enlarged spectrum beta-lactamase, which are really becoming a big problem in Europe. And important statement here for Phage Espoir. Phage Espoir actually it's very important. They are going, as Jérôme explained, they are starting to build a patient clinical register, and we decided that all the patients that are going to be included in the trial are going to get in this patient clinical register. And because this is an international clinical trial with France, Belgium and Switzerland, we are going to try to do that also for the other patients that are going to be treated in these countries outside of the clinical trial. But if you have some ideas and if you want to contribute to this patient registry form from your own countries, please get in touch with Jérôme. I think we need to coordinate these efforts. Just a quick slide about the products. So as Marta was explaining at the beginning, Pherecydes Pharma started to do some work on recombinant phages. And actually, when we went to discuss with the clinicians and said, oh, look, we have nice recombinant phages that we could use for treating the patients, it's not only the issue of virus, the scary virus, it's also the issue of coming up with GMO treatments. And I can tell you that in Europe, if you do that today, especially GMO treatments that you give to a patient, it goes out from the treatment, sorry to be a little bit vulgar about that, pees on the sidewalk and release some genetically modified viruses, I can tell you that none of the clinicians want that. So we switched totally the strategy of the company in 2010 to really go to natural phages. What we have done, we have been screening about tens of samples of wastewater, whatever. We have a collection of close of 1,000 bacteriophages against E. coli. They have been screening against about 300 different strains of E. coli, representing the genetic diversity of the species. And also we collect regularly some strains coming from different hospitals, including multi-drug resistant strains, and we have a lot. And then we started with 23 pre-selected phages. The product is now 16 phages that have been produced. They have been tested into a GLP mice model with third-degree burn, multi-drug resistant infection, one strain which was only still tolerant to 2 antibiotics. And on top of that, according to the clinicians that from the hospitals, we also included immunosuppression because all these third-degree patients are immunosuppressed. In that model, the animal dies in about 2 days. And with a phage cocktail of 16 phages, we saved all the animals, one single shot. We did dose effect, we did pharmacodynamics, and we have excellent data. And we just finished the mini-pig study with SC and IV. The safety is complete for that product, and the second one is about 2 months behind. I'm going to come to the conclusion quickly. I want to really thank here 3 people that have been key in this project. Here on the right you have Dr. Jo, who is the chief of the burn unit in Percy in Paris. Here you have François Rava, who is the chief of the burn unit in Lyon, in France. And here you have my R&D director, Flavie Pouillot. Without them, we wouldn't be there. And if you need to talk with us during the meeting, there are 2 representing persons of the project, both Jerome's, so you know us. You can come and talk to us. Thank you.
Questions? How big will the trial be, and will there be any healthy volunteer work or straight into phase 2?
Well, we got some kind of an approval to go straight into a mixed clinical trial, phase 1, phase 2, combined trial. And the size is rounded about 200 patients.
And how are you going to administer the phage to the patients?
We worked with the nurses on the hospital and we checked how they proceed usually with the standard bioantibiotic treatments. And actually the way they go for third-degree burn patients, they use bandage that they soak with some antibiotic liquid solutions. So we went and see how they were doing, and we decided to prepare a formulation, liquid formulation. Because also one of the issues that I should mention, we suspect that there is a risk to have, when the product gets on the market, quite a bit of off-label use. Because of course the E. coli product, the E. coli cocktail, we have, for instance, many strains that come from skin infections, but the vast majority actually has urinary tract infections. And we know we have a product which is 95 to 96% efficient against all the urinary tract infections we have been facing. So we suspect that some of the clinicians probably will want to use that as a last treatment.
They'll receive systemic antibiotics as well as topical phage.
Yeah, we get into the details of the clinical trial. Actually, it's a process where we go 2 days before patient inclusions. You have swabs being taken in a specific manner, which is going to be exactly the same in all hospital burn units. Then you do a control test, and all the analytical bacteriology lab can do that in less than 48 hours. And if the patient has an E. coli, whether sensitive or resistant, it gets into the trial. Same thing for Pseudomonas. Now, if during the process of the treatment, secondary strains come up, of course, we have included the possibility to add secondary antibiotics if you end up having a Staphylococcus aureus treatment.
Will the phages be in a hydrogel on a dressing?
Liquid, simple liquid formulation. Yes?
Are you going to depend primarily on wound cultures, or are you going to do PCR? Are you going to do a microbiome of your wounds so you really know what's there, or are you just going to do simply what cultures out?
I don't know. The bacteriologists know that. I'm not in the detail of how this is going to be analyzed. No other question? Thank you.
Thank you very much. Best luck.